I actually wanted to talk about being useless, and then I realized that this was also a place to talk about failure. So I decided to change the title in my talk from the one that you see in your brochure to one that I think adequately describes me, failed and useless. And I want to talk about both of these issues and bring them up in the course of the next 40 minutes or so. But first, I want to give some thank yous. The first, obviously, is to the UPenguin Charitable Trust and Dennis Pang for supporting this effort that now I'm a beneficiary of, and supporting actually the more long-term goals of the EPIC initiative that I think are very worthwhile. So I applaud the foundation for their insight in this support. And as you see at the bottom, I've put Zhao Dong and Stuart. Stuart is a colleague of mine, but he's also the deputy director of EPIC, and I want to thank them for getting me involved in all of this and being able to participate. So I've heard already, both at lunch and a little bit today, about this idea of overcoming failure. So I'm not sure I'm the right person as a role model of this. I've certainly had some not really great situations, but I'm not really sure I'm a role model. These are my grades from college. And I want to remind you that I have a Nobel Prize in chemistry. If you look at my grades in college, you will see that there is nothing. So let me tell you a little bit of the narrative. I go to college, I take some math courses, which I really like. I get fairly good grades in them. I then, in my sophomore year, take one more math course and realize I am no good as a mathematician. I go searching for other things to do. You can see there's not a single grade above a C in chemistry or physics in my entire transcript. So I became a biologist. Actually, that's not true. What I really did is I was a biochemistry major, which allowed me to take all these different courses. And I went to work one summer because undergraduates get opportunities to work in labs. I thought this was a good idea. And I had lots of bad ideas about who scientists were, which I'm going to tell you about in a moment. But I decided I would go work in a lab. I got into the lab a little bit late. It meant I couldn't be in the room with everyone else. I had to be in a student lab 50 yards away. I was all alone for the entire summer. I had no idea what I was doing, but I had decided that real scientists never ask anyone for help. They have to do it all themselves. And so I didn't ask anyone for help. So it will come as no surprise to you that the experiments never worked. Actually, I think one or two times they worked, but I didn't realize they worked. So I failed completely at this. And as a result of that experience over the summer, I decided I am never going to be a scientist. So I didn't overcome failure. I just succumbed to it completely. Now, I should also point out one other thing about grades that I want to say here. Our chemistry department over at, across the street, the people there say that the thing that annoys them the most is that students come in, the very first science course most students take is general chemistry. And the faculty is appalled by the fact that every single time a person gets a B, they immediately drop the course and decide to major in economics. But what they really do is they're taking the grade as the image of their self-worth and everything. And I certainly did too. So I had not very good grades. So I shouldn't be doing any science. And I didn't. After I graduated from college, my senior year, I took all these wonderful courses in the humanities and social science where I learned a lot. It was a lot of fun. But afterwards, I worked for 1 month as a janitor, 1 month in a factory. I then studied sociology, thinking I would be good at that. Failed at that completely. 6 months after that, I decided I wasn't going to do that anymore. Then I interviewed hospital administrators, another 1 month job. My father got ill. My parents owned a dress manufacturing company. I went on the road and sold dresses for a month. And then I had the summer off, so I arranged rock concerts for a nonprofit in Cambridge, Massachusetts, and then got my first job. And my first job, real job, was teaching high school. At a private school outside of New Haven, and I taught chemistry and algebra and social science, and I was there for 2 years. In the middle of those 2 years, I made an astonishing discovery. In the summer, students have summer vacation. Teachers have to find a job. And so I was looking for a job, and I was exceptionally fortunate because a friend suggested that a researcher in the ophthalmology department at Yale, this man, Jose Zedonaisky, was maybe going to have a place in his lab and I could work over the summer. So I went and I talked to him, and he told me about his experiments with frogs, and it was about a problem that is, I still find astonishing to this day. It shows how I wasn't thinking about the problem. The minute I heard about the problem, I thought it was one of the most wonderful problems I had ever heard of. So I'm going to do a little test here. What I worked on was the transparent part of the body. Now, without saying anything, if you know what the transparent part of your body is, raise your hand. Most people will not raise their hands. I already talked to Stuart about this. He already knows. But he also did. Well, let me give you a hint. The transparent part of the body, it's right in front of your eyes. It's your cornea and the lens. But I have never met anyone, even people that say, oh yeah, it's the eye. I've never met anyone that said, you know, I've always wondered why that is transparent. What makes it transparent? And that's what this man was studying. So I was fascinated simply because I never even thought to ask that question. He told me about the work with frogs. He said it was about chloride transport that was needed for this. And there was an apparatus that he used called an Using chamber in those experiments. And I remembered something from when I was an undergraduate. I had read a paper and written a report about a paper that also used an Using chamber. But to just tell you how completely wrong I was, although they used the same apparatus, it was not working on frogs, it was working on toads. It was not the cornea, they were working on the skin. And it wasn't chloride moving, it was sodium moving. So everything was wrong, except they used the same apparatus. This is like saying, oh, they wrote that in pencil. I know another thing to write something in pencil. And so, but I remembered this, and I remembered the conclusion that a small molecule called cyclic AMP turned the system on. And so in the middle of my interview, I said to him, does cyclic AMP work here? And he said, I don't know, but you're the second person in 2 days to ask me that question. And I think because I asked him a question, he hired me, which I consider the first miracle. He then gave me an absolutely boring topic to study for the summer. And then the second miracle occurred. He walked into the lab and announced that he was leaving for the summer and that he was doing his own experiments in France. Another reason to want to be a scientist. In any case, he was gone. But there were people, that other person that asked the question, other people I could ask, and so I asked them, how would you test this? What would you do? And one person said, I would throw otherwise called epinephrine. I would throw that on the system and see what happens. And so I did that experiment. And you can see where I added the drug, and boom, the whole thing got turned on. I spent the whole summer doing those experiments. When the guy came at the end of the summer, he wanted to know what happened with the boring experiment. And I said, sorry, I didn't do that. He looked a little surprised at me. And I said, but I do have this, and I could go to the library, and here's the literature I found out about this. And this became my first scientific paper. Basically, what it said to me was, I sometimes can come up with ideas, even if the origin of those ideas is sort of silly, that I actually can get an experiment sometimes to work. It gave me a lot more confidence, and it allowed me to then think about going to graduate school. And I got to tell you, having something work is a great stimulus. Okay, so why did I have all these had these incorrect ideas about science? Well, I think it's because of all the stories I heard about the great scientists when I was growing up. So what were those things? You'd hear about Einstein and Newton and Galileo and Fermi. You'd hear about all these wonderful scientists, but the stories, the way we tell them, I think, is incorrect. So what were those incorrect ideas? Well, the first thing is that we talk about these people and the amazing work they do. We give everyone the impression that they're geniuses. And if you're not a genius, for example, you get C's in chemistry, you shouldn't do this. So I had that idea that really good scientists had to be geniuses. The second thing is that scientists' experiments work all the time. And the reason they work all the time, we don't have time to talk about them failing. So we just get right to the answer. We never say how they've gotten to where they are, which is why I'm excited about this EPIC initiative. The next thing is that scientists think differently from everyone else on the planet. They use something called the scientific method. They come up with a problem that probably no one has thought of before. They then think about that, develop a hypothesis, test their hypothesis. The experiment works the first time, and they just get a lot of prizes and honors. But they're very purposeful in what they do. The other thing about these scientists, and I should tell you that when I was in grammar school, people didn't really teach elementary school children about DNA, Watson and Crick. So as far as I was concerned, every scientist I ever heard of was someone who worked alone. Now, they could have an assistant, and I don't know why, but for some reason, the assistant's name had to be Igor. But they always worked alone, that this was a solitary activity. And the final thing about hearing about all these great scientists from the past is that you get the idea that, except for some very rare exceptions, George Washington Carver, Marie Curie, every single one of them are European or of European descent. They're all white men. And that's the image. Those were the scientists we were talking about. And if you think that's not a problem today, it certainly is. If you go online and you ask for images of famous scientists, this is the top group of people that you get. And only in here are Marie Curie at the top and Rosalind Franklin at the bottom. You have to go to the next panel to see George Washington Carver. Everybody is European or of European descent. In contrast to that, I want to show you the picture of what my lab has looked like, the people that have come through my lab in the last, 8 or so years. We have people from Chile and Canada and Peru and China and Iceland and Greece, the Gambia, Vietnam, Israel, all over the world. This is a characteristic lab. Science is a universal occupation, and we have to keep it that way. Okay, so those are all the bad ideas. I'm going to give you some examples in a little bit about how we can maybe get over those bad ideas or what the real truth is. And I'm going to do that by talking about the discovery and the work that I was involved in this wonderful protein called green fluorescent protein. But before I do that, I want to talk about uselessness for a little bit. And so by uselessness, I actually take this from a wonderful article that was written 1939 by Abraham Flexner. Abraham Flexner was an amazing individual, an amazing educator. He was the person in 1910 who chaired the committee that came up with our present-day guidelines for education of pre-med students. All the pre-med requirements came from the Flexner report of 1910. And then, as I'll show on one of the slides, another little thing that he did was to convince people, a donor and then people at Princeton, to have the Princeton Institute for Advanced Study. And he was the first director of that. Anyway, in 1939, he wrote this wonderful article called The Usefulness of Useless Knowledge, which he says, from a practical view, intellectual life is on the surface a useless form of activity. In this paper, I shall concern myself with the question of the extent to which the pursuit of these useless satisfactions proves unexpectedly the source from which undreamed of utility is derived. And he starts off with a wonderful story. He says he met the business leader, George Eastman Kodak, and he's talking to him. He says, who do you think has been the most influential scientist? And Eastman, without any hesitation, says, oh, it's Marconi in the development of wireless and the radio. At which point, Flexner says, whatever pleasure we derive from the radio, or however wireless and the radio may have added to human life, Marconi's share was practically negligible. He was inevitable. The real credit for everything that has been done in the field of wireless belongs to Professor Clerk Maxwell and Heinrich Hertz, the people that discovered the science behind that enabled Marconi to do this. And to just, and those two people are shown there. And then I happened to be reading a whole bunch of quotes by Mark Twain, and I found this wonderful quote by Mark Twain. Whoops, right, come on, there we go. Sorry, hit it too many times. Should go back. There we go. It takes 1,000 men. A little temperamental, but we'll get there nonetheless. There we go. It takes 1,000 men to invent a telegraph, or a steam engine, or a phonograph, or a photograph, or a telephone, or any other important thing, and the last man gets the credit, and we forget the others. And it's all those thousands that I think are important, because those are the ones doing the useless work, getting the useless knowledge. And to bring this to a more modern example, there is this wonderful poster, which you, I know, cannot read on this slide. But I just want to show you the whole extent of the poster, and it can be gotten from quantsoft.com. They made this poster in 2014 to show what was needed to make the iPhone. So here's the iPhone. That's the culmination of this, and there are various things, wireless, LEDs, computational things, and so on. And they went back and said, who discovered these things? What needed to be done in order for there to be an iPhone? And so here's one part of the chart near the top, where you can see all these amazing people doing it. But I want to point out what's at the bottom. Down here, in 750 BC, the first lens is made. And in the first century, up at the top, it says the first magnification. So the lens for the camera started that long ago. And then on the left-hand side of the slide, there are things about computation, the first use of binary, and so on. All of these things are needed before you could get something that is the iPhone. So let me tell you my story, or my involvement in, and that shows some examples of failure and things that I hope I can convince you all those things I learned about scientists are completely wrong. So the story for me starts off when I happened to be sitting in a seminar, and the seminar speaker is talking about this man, Osamu Shimomura, and his work on this jellyfish, Aquaria victoria. Osamu Shimomura shared the Nobel Prize. Unfortunately, he died last October, and had a remarkable life. He was in Nagasaki, or he was sent just soon before the bombing of Nagasaki, outside the city, and so he was protected when the atomic bomb destroyed the city. He could not go to college for several years. He eventually was able to when they rebuilt the college. The first college they built was pharmacy. He didn't want to go to pharmacy school, but it was the only college he could go to. He went to work at Nagoya University as a technician. He was given a project that several other people had failed at. He got it to work. As a result, he got an invitation to come do more work in the United States, and he got a rather unusual going-away gift from the head of the lab, who arranged for him to be given his PhD. Not a bad going-away present. And I had known about some of his work, but not all of it. What he was interested in was another fundamentally important scientific endeavor, trying to understand why different organisms can actually generate light. We have fireflies and glowworms and bacteria and fish and all sorts of different organisms can make light. That's called bioluminescence. And his first work at Nagoya was on this. This became his life work, to understand why different organisms in many different ways can actually generate light. And he came to the United States to work on this jellyfish. This is where failure and the scientific method come into the story, because he spent the whole summer getting jellyfish, isolating the cells that normally should produce this green light that the jellyfish does, and every time he did the experiment, he got nothing. He's a very good biochemist. He failed every single time he did the experiment. Towards the end of the summer, he had failed many times, and one night he finished with another failure just at the time. It was dark outside. He wanted to go home and have dinner. So he cleans up after himself, and he throws all the samples in the sink. Now, the sink also had some seawater and probably some other junk in it too, but he throws it in the sink, turns off the light, and is about to leave the laboratory when he looks back at the sink, only to see that the sink is glowing brightly. He thinks about that for a little bit and says, maybe there was something in the seawater that I didn't have in my reactions. He thinks about that for a while and says, calcium. Goes back later, next couple of days, repeats the experiment, but this time he squirts in a little solution with calcium ions in it, and every time he does that, he produces light. Complete accidental discovery. Now, I have heard of people making such similar discoveries by throwing things on the laboratory floor or on their lab bench, collecting them up. Not exactly the scientific method. Adds calcium, and he names the protein he purifies aquarin after the jellyfish, and it produces a beautiful, intense light. The only problem with the light is it's the wrong color. So he looks at this thing and he says, it's blue, it's supposed to be green. Maybe there's something that converts the blue to green. Now, molecules that have an input of one color and an output of another, those are fluorescent molecules. And sure enough, he finds another protein that all you have to do is shine blue light on it, and what you get out of it is green. And this is he called it the green protein. We call it the green fluorescent protein. He wrote about this in 1962 in a footnote to his paper. As far as I know, it's the only footnote to win a Nobel Prize. But aquarin plus calcium, now with GFP, gives the green light that he was looking at. So 2 accidental discoveries. Throw it in the sink and get the wrong color. But I'm listening to this talk and I realize you don't need the calcium, you don't need the aquarium. All you need is blue light. It hits the molecule, you get green light out. If I could put this protein into the cells that I was studying, I would be able to see them by shining blue light. And only the cells that had this protein would shine back green. And I got really excited, so excited I did not listen to the rest of the seminar. I have no idea what the speaker actually talked about. Because I realized that this was going to be able to provide a lantern. And I was very excited about this. And I was able to collaborate with someone who was isolating the DNA for this, because we needed to put the DNA into cells and allow the DNA to be the instructions to make the protein. And I had a wonderful graduate student, Gia Skirkin. As I say, I worked with Douglas Prasher, who was doing the DNA work. And he sent us the DNA. And the first experiment that Gia did was to put it in bacteria. And this is a page from her lab notebook 1 month after she entered graduate school, where she found that there were very strongly fluorescing bacteria. We were very excited about this. We now realize we could put this anywhere. It didn't need anything extra. But the other thing that I like to point out about her page from the lab notebook, Gia had already gotten a master's degree from our engineering department, working on fluorescence. She was the perfect person to do the experiment. When she got her bacteria, she put them under our microscope, but my microscope was a piece of junk. And as a result, she wasn't sure if it worked or not. So she went to her old lab, and that's where she confirmed it and was able to take this picture. It left us with a problem. That's what it says here. She said she worked in engineering to do this. We didn't have a microscope that worked. How did we solve this problem? I called up all the sales representatives of microscopes in New York City, and I said, we have now discovered a new way to look at genes and cells and everything. We need a fluorescent microscope. I'm absolutely going to buy a fluorescent microscope, but I don't know which one to buy. If you would bring yours by the lab and let me try it out, hopefully for a month or two, I think I could give it a good working out and we'd be able to do this. And they did. So everything was done on borrowed microscopes, and then we finally did buy one. But GFP, or green fluorescent protein, has been used in many, many different systems. Here's a little rogues gallery of different organisms and cells. Up in the upper left-hand corner is the worm that we work on, and what you're seeing, the worm only has 302 nerve cells. That's its nervous system. Every one of the nerve cells is in green. The rest of it we don't see. And the basic idea is, once you can see something, you can study it. And so these are the cells we really study in the animal, and there's lots of questions that we could now ask that we were never able to ask before. So for example, can we find mutants, variants, that are different from the normal situation of these cells? And to give you one example, here's one. If you look up in the middle, I actually have a pointer here. If you look up in the middle, you see this cell right here. That only goes towards the front, towards your left. Whereas in this mutant version, it grows a new process that actually comes out the back. So we have mutants that change the shape of cells, the position of cells, the number of cells, and each one of these mutants allows us to study those aspects of how normal nerve cells grow and develop. Without having this way of looking directly at the cells, we would never be able to do it. And more than that, we can look over time. Another thing that we are working on in the lab, this time using not only GFP, but a red fluorescent protein, is the fact that we start off with our cell, which I hope you can see this little green dot here, and it's right next to this red, which is muscle. But as the animal becomes an adult, the green separates from the red, and we can see it by putting GFP in the nerve cell and RFP, red fluorescent protein, in the muscle cell. It's very easy to see that gap. And so we simply said, are there mutants that interfere with this process? And the answer is yes. We can get animals in which the cell never leaves the area next to the muscle. And we can start to study this basic problem of how tissues interact and how cells become encheathed by other cells, simply by looking for mutants where this goes wrong. So this has been why I think GFP has been used enormously for many, many different ways. And I'm going to divide it up because I think every discovery in basic research leads to more understanding of basic research. It leads to applications that have more direct bearing on human activity, and they come up with things that are truly unexpected. So let me give you one example. Normally, if you look at any textbook, and look at a picture of a cell, you will see that in the cell, there are bits and pieces, but most of it is in a soup called the cytoplasm. And no one really cares much about that because it's just what contains everything else. Well, Cliff Brangwyn, a postdoc, not in my lab, in Tony Hyman's lab, looked at GFP labeling a protein that was supposed to be in the cytoplasm. It wasn't in the cytoplasm, but it was in special components that acted as if they were like oil in water, that they were separated from the rest of it. This has now become an incredibly hot area of research because we've suddenly discovered that lots and lots of parts of the cell have this division, what's called phase separation. And in fact, some people think that maybe the extreme of phase separation is when certain proteins come out of solution. When does that happen? Things like Alzheimer's disease and others. And that maybe understanding this thing this was in 2009. This was after our Nobel. So this was a discovery of a completely new way of looking at the cell that may have lots of implications. Another experiment done around the same time was to ask the question, how does the AIDS virus go from one infected cell to another? So people at Mount Sinai here in New York said, okay, let's make a version of the AIDS virus, HIV, and whenever that infects a cell, the cell will become green with green fluorescent protein. And we will be able to see it transfer to another cell. Now, most people are taught in college, viruses spread from one cell to another because the cell explodes and then another cell picks it up. But if that's the case, if it explodes, it's out there. It could be attacked by an antibody. It could be attacked by other cells. What did they find? Well, what they found, here's the picture. There's the infected cell, and there's a little particle of virus right at the tip, and there's a cell that's about to be infected. No explosion. It just gets transferred into the other cell. This means that if you want to look at a way of controlling the spread of this, you have to start looking at how the transfer is made. They were able, this is in mouse cells, this is not done in people, but this is looking at how this sort of transfer can be done. And then there have been people that have done all sorts of wonderfully inventive things that I just think are spectacular, that I would never have imagined. One of them is a guy named Bob Berlage. Bob Berlage made what you might think is an incredibly silly idea. He knew from work of others that there was a controlling element in the DNA that would turn on a gene in the presence of TNT. And so he made bacteria that whenever they were in the presence of TNT, the explosive, that bacteria would start to make green fluorescent protein. And you might ask yourself, why would somebody want to do something like that? Well, his goal was he was trying to figure out a way to be able to detect landmines, because landmines leak TNT. And so he took his bacteria, he was at Oak Ridge national laboratory at the time. They were able to get some landmines that they disconnected. These are not stupid people. They disconnected, but they still had the TNT in them. He had a colleague bury them in a 3-meter by 5-meter plot of land, and then he went in at night and he went in during the day and sprayed the area with his bacteria, came back at night with an ultraviolet lamp, and was able to see where the landmines were buried. Now, unfortunately, this did not work all the time. I know of at least 3 other groups that are still working on this, because unfortunately, this has to work 100% of the time. But the idea of using this to counteract one of the most egregious aspects of war that kill bystanders and innocent people, often well after combatants have left an area, is, I think, astonishing. So the applications are all over the place. I should also say that GFP has been used as the explanation in the Ang Lee movie Hulk of why the Hulk has green skin, but I won't go into that. What I want to go into is what I talked about, do we have a counterexample? So our scientists, geniuses. Well, Roger Chen, the 3rd person that shared the prize with us, there's a very prestigious science talent search for high school students, and he won as the best high school science student. So if anyone from a very early age was doing spectacular work, it was Roger. I've already shown you my grades, so I think the conclusion we have to draw from this is that some might be geniuses, certainly not all, and for none of us all of the time. And I'm not going to explain that at all. The next question. Scientists' experiments work all the time. I told you Shimomura's story, so rarely. So they don't work all the time. Scientists use the scientific method. They do, but I believe that they do it after they make a discovery. Enrico Fermi had this wonderful line that Stuart told me about, in which he said something on the order of, if you do an experiment and it confirms your hypothesis, you've made a measurement. But if it doesn't confirm your hypothesis, you've made a discovery. So those discoveries come because you're aware of what's going on, you try to keep your eyes open, try to think about the things, but then you say, okay, well, if that's the case, now what do I do? And you think about things. So I think the scientific method comes after the discovery. Okay, next one, scientists work alone. Well, these are all the people that were on the GFP paper. Every one of them were important for the work. We definitely did not work alone on this. So again, that one you'd have to say rarely. And this last one, scientists is done by white men, just nonsense. Now, I also want to hammer in a little bit more about the uselessness of basic research. William Proxmire was senator from Wisconsin, used to have a famous press release called The Golden Fleece, in which he tried to describe government-funded research or government-funded activities that he thought should be belittled. He did this for many years until one time he belittled some research that actually gave an enormous benefit to people with diabetes, and then he apologized. A few years ago, 4 members, 2 Democrats and 2 Republicans in the House, decided that they wanted to have a different award. They did this with Science Magazine and other organizations, and they called this the Golden Goose Award. The award highlights and honors examples of scientific studies and research that may have seemed obscure, sounded funny, or which the results were totally unforeseen at the outset, but which ultimately led often surreptitiously to major breakthroughs that have had significant societal impact. So I was very fortunate. They decided GFP was one of those that they should get. So I got this. But I want to talk Charles Towne, who developed the maser, which led to the laser. That was given an award. people that had researched the idea that coral would be a great surgical substitute for bone and is used all over. And the one I want to do is this third from the bottom, rat and infant massage. Some people were looking at rats and they were separating the newborn rats from their mothers, discovered that the rats absolutely didn't thrive when they were separated, put them back in and watched what the mothers were doing. And what the mothers were doing was licking the rat pups. They decided to do an experiment. They did not lick the rats, but they did take their fingers and stroke them. And when they did, those rats separated from them still thrived. This now is used as a technique for premature babies to get them to thrive as well. So a chance observation, follow through on it, and then an unexpected and it wasn't purposeful in any way to do this. Really quite wonderful. So I'm probably over time. Is that right? Okay, so let me end by saying, giving you one last quote, which is my favorite quote about basic research and the importance for basic research. Let me first say that I'm not thinking about it as to replace applications. I think engineering applications, medical applications are exceptionally important. As I get older, I think they're more and more important. But I really think that in order for these breakthroughs to be made, we have to have a pipeline, and that pipeline starts from observations in basic, or discoveries in basic research. I think there are people that are going to be very good at applying things. I can also tell you I don't know a single basic researcher that does not think to him or herself about the implications of whatever discovery they made. There is no problem of people wanting to apply what they learn. Sometimes we get this feeling that people are pushing, oh, those ivory tower scientists don't think about the implications. Completely untrue. But we need to continue the pipeline. So I want to leave you with my favorite quote about basic research and its importance. And this was done by the first head of the Fermi Lab, which until the super collider in Europe came on board was the largest particle accelerator in the world. It was Robert Wilson who said this, and let me quickly go through these slides that I now no longer have time to do. They're much too long a talk, I'm sorry. This is the quote. He was asked by a senator, what's the advantage for national security for doing this big science project? What about the national defense? How will this help national defense? And this was in 1969, so right in the middle of the war in Vietnam. And Wilson said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
I'm very excited to be here, and thank you for your wonderful words and wisdom about science and failure. I want to start by asking you a question about how we should perhaps interpret failure, because sometimes I think failure sends a message, and it's an important message. It could mean you're doing the wrong thing. So how do we listen to failure without judgment, perhaps, but extracting the important information out of failure?
So I'm not sure, because every time we face failure again, I get depressed. But I think that part of the answer is that giving up is a problem. Mm-hmm. Okay. I have an answer. I often give talks that I talk to graduate students, and they ask me, how do you cope with failure in the lab? Which obviously means they feel a lot of failure in their graduate work that they're doing, which I think is sort of understandable. And I give them an answer that I tell them right off the bat they're not going to like. but it has helped me enormously. And what I usually say is, you know, you can find in almost any lab people that are unhappy because experiments have failed. But it's very rare that you find the head of the lab upset about that. So why is it that the head of the lab doesn't seem upset, but the people in the lab, some of them can be upset about having experiments not work? And it's not a nice answer, but the answer to me is there's more than one person working on a project in the lab. And so the head of the lab likes who's succeeding and the work that's going on, because that can proceed forward, knows the others are going to come into it, but has the opportunity because people are working on multiple things. And I suggest to students that one way of overcoming a feeling that you're hitting your head against the wall is work on more than one thing. Not exactly what anyone wants to hear. You're failing, do more. But I think the idea of using more than one, having more than one goal. In my work, I work on a set of nerve cells that sense touch in that tiny worm that I showed a picture of. Now, some of the mutants that we work on are mutants, they're touch insensitive because the cell isn't made. Okay? In others, the cell is made, it just doesn't work. Well, those are two completely different avenues of research. The first one is asking the question, or addressing the question, how do you make a particular type of nerve cell? That's a developmental biology question. The second one, well, it's made, but it doesn't work. How does it work? What are the components that make it work as a sensory cell? Two very different questions. And sometimes in the lab, we're working on one because that's where the progress is being made. At other times, we're working on the other. And they shift back and forth all the time. So I think part of addressing it is doing more than one thing.
Not failing all the time.
Yeah.
So when is it okay to say this failed? I mean, sometimes we just have to say this did not work, this was a dead end, and now we have to do something else. I mean, it's not always wise to keep brushing yourself off and do the same thing over.
Yes, no, I completely agree. And I don't have any real rules for that. I think if people put a bona fide effort in, let's say a graduate student or a postdoc, put a bona fide effort in, could be 6 months, but having tried many things, that may be enough to say maybe we should now direct your work towards a different goal, knowing that they're always going to keep that other thing in the back of their head and thinking about it if something else comes up that gives them some insight into the problem. But it's I think this is one of the biggest problems of trying to decide when it is enough to stop self-torture. This is, but I think there is, because I felt it myself, when things don't go well, the last thing you want to do is, the last thing I would want to do is go back in the lab and try it again. It's very hard to brush yourself off and do it another time. I think you have to somewhat force yourself to do that, but I don't have any general rules.
I want to ask you a little bit about expectations. And I guess this is a broad question, but failure is often tied to our own expectations or to what we want to accomplish. And I recently interviewed a scientist who works at the Wistar Institute in Philadelphia, and she studies how cancer metastasizes. And she says, if I can figure out in my lifetime the mechanism of how this happens, I will have done an enormous part of what I wanted to accomplish. So she wasn't thinking about curing this cancer. She wasn't thinking anything like that. So she will probably not walk away from her career thinking she failed. But I think sometimes scientists think that they will do the next big thing, and then just being part of the journey perhaps feels like failure on some level.
Maybe part of the problem is the way we tell the story. That, I don't think anyone here wants to hear about our daily successes and failures in the lab. I wouldn't want to hear about them either. So I think those things, but we actually do make those successes. I mentioned to you before, I actually think that failure is a very necessary part of science. And the story I like to say about that has to do with my daughter, who's now 26, but when she was 12, I was inducted into the National Academy of Sciences. And the ceremony, if any of you have trouble sleeping, get a video of the induction into the National Academy of Science. Each member that's newly elected gets on the stage, shakes the hand of the president of the academy, has a picture taken with the president of the academy, walks across the stage and signs a book. They're told everyone has signed it, so you think, I'm signing the book with Einstein and all these wonderful people. And then they say, well, actually, we started the book in 1970. And you do that, and you walk down. And while you're walking across the stage to sign the book, someone reads one or two sentences about your work, why you were elected to the National Academy of Sciences. And as I said, my daughter at the time was 12 years old. I knew she didn't have a lot of patience, and I was feeling very happy that she was there, but sad that she had to sit through an hour of this stuff. So when I finally was able to make my way down to my daughter, where my daughter and wife were sitting, I said, sweetie, I'm really sorry, I apologize, but I'm very happy that you and Mom were here to help me celebrate, but you must have been bored out of your mind. And she looked at me, and she often looks at me like I'm the stupidest person on the planet, and says, Dad, don't you understand about those people? I played along. I said, what do you mean? She said, because she had been listening to those sentences. She said, Dad, each one of those people were the first person in the world to see or do what they did. Wow. I'm exceptionally proud of that answer. But if we're always trying to be the first to see something or to do something, I believe failure is an inevitable part of that because we actually don't know what we're doing. If we know what we were doing, we'd just solve it. That would be the end of it. We're trying to find things out that we do not know. And so failure is actually a part of this process of discovering nature, discovering the world around us.
Mm-hmm. I'm wondering if you could give us an example of a more mundane failure that you experienced perhaps recently. So not your failure to get an A in chemistry, but something that happened in your lab that really frustrated you to the point where, maybe you wanted to throw things in the sink.
I wish I had things that I could throw in the sink that would give me those results. That would be great. So often I will construct ideas. I say, well, if this is true and then this is true, then if we do this and this is going to work. I know it's going to work. Let's see what happens. It doesn't work. You throw out your hypothesis. You do something else. So I can't think of a direct one right now, but we do a lot. So, for example, we're doing some work in which we're looking at bacteria that are interacting with these worms. And we wanted to know how were they doing it. There had been a previous paper working on a more remote thing than what we were working on, but they had a certain idea, certain pathway. This gene product works with this one, works with this. We said, oh, great, it's going to be the same thing. It wasn't the same thing. Now, that's wonderful because now we can do something new and different, and that's good too. But we had the hypothesis, it fell apart. This happens all the time. I mean, I don't cry. I don't scream at the things. I do scream sometimes. I scream when the reviews come back from our papers and they want us to do more stuff. That's when I scream. But that's a different issue entirely.
What helps you get a fresh perspective? If you've had a frustrating day, do you take a walk? What do you do?
So I get my ideas, I think, in 3 or 4 different ways. One of them is talking it out, diagramming the experiment, talking with people in the lab. So many things have come because we have a little bit of information, we try to think out the thing, and it's in the actual talking of it that we develop ideas. I also have been fortunate to be able to travel a lot, and I go in, part of visiting another university is to go in and talk with their individual scientists, and they'll talk about their work. And it'll be something I've never heard of before, but in quite a number of times, I could say, oh, I wonder if this is happening here. And as I said, that's how I got the idea from listening to this talk about Shimomura. Yeah. What I didn't say in that talk, which I should have, is that the animal that I work on is transparent, this tiny little worm. And so when I heard that there was a way of shining a light or having a light turn on within the organism, I was very excited. I know that no one else in the room got excited by the talk in this way, but they didn't work on any animals that were transparent. And in fact, I was the only one in the room that was asking the question, where are genes turned on? And I realized this was a way of being able to see it, and most importantly, I'd be able to see it in living animals, to be able to study living animals. So sometimes it's just these random inputs because there's all this nonsense that's still swimming in my head of all the other failures that we have or things that we'd like to see. And then it comes up. So it's actually interacting with people or papers or talking about particular experiments in the lab. It's the interaction for me that's important.
This is a question that came in from the audience. How do you work with your students in terms of dealing with failure? How do you talk to them about failure? How do you encourage them? How do you make sure they learn from it?
So in part, we do talk about it. We talk about trying other experiments, trying other things. There comes to be a point when we actually say maybe we should try a different tack, maybe we should try a different set of experiments. I recently had someone talk to me, very frustrated in the work, and we sort of talked, again, it was one of these cases of talking out the experiment, and it turned out there were several things that still were pretty exciting about that to try in slightly different directions, and that sort of helped me and helped this member of the lab to also sort of address these things. So again, it's mainly talking about it, recognizing that these things don't work all the time. I'm not a very good person for troubleshooting experiments. Saying, oh, you have to use twice as much salt in this solution, or you have to, this is missing. I know a little of that, but not a lot. I actually interact with my students, my postdocs, by having them first choose a project they want to work on, not assigning them a project, and just trying to be supportive, making sure they have the resources and can contact people that can help them put that together, and being excited about their research, and we talk all the time. So, but I'm not a person that says, okay, now you have to do this experiment, or now you have to do this, or here's the next experiment. I will suggest things periodically when I get too excited to keep my mouth shut, but I do like people to sort of make these discoveries on their own, and they do.
Yeah. Here's another question that came in. So winning a Nobel Prize is a dream for many people. How did you reconfigure your goals after accomplishing this? In other terms, what does success mean now since you've won that prize?
So a couple of things. The work, I heard this seminar in 1989 through a problem of keeping in touch with Douglas Pressure. We weren't able to do the experiment until the fall of 1992. We published the paper in 1994, and we basically never did too much work after that on the topic. So I was very fortunate to get a Nobel for a single paper and something we did in a very short period of time. Before that, I was working on nerve cell development and function. After that, I was working on nerve cell development and function. Then I got a Nobel Prize. I still work on nerve cell development and function. So it didn't change the direction of the lab, but it has given me a lot of wonderful opportunities. I get a chance to meet more people. One thing, as you've all heard today, I hope you liked it, is that scientists are usually not asked to give general talks. Nobel laureates are always asked to give general talks. And I enjoy that a great deal. The other thing is I am now, in a sense, on a list. I get asked for things, and most things I don't do. But one thing that I was asked to do, and I'm exceptionally proud of it, is at the National Academy, there is a committee on human rights. And they asked me first to join it, and then they asked me to be the chair of the committee. And I've been the chair of this committee now for the last 4 years, and it's exceptionally rewarding. We work on behalf of scientists, engineers, and health professionals who, anywhere in the world, including the United States, who have had their human rights abused. And we do a number, we have symposia that we design, I go on embassy visits. There's a number of things we do, and I found it exceptionally rewarding, and it's a direct consequence to the fact that I have this Nobel, and so they considered me for that position, and I'm very, very happy about that. Yeah. I should also very briefly say that there are a lot of things we think Nobel laureates get. They do not. Okay? And the reason I think is because we view Nobel laureates, the ones we grow up learning about, as being at the top of our field. And so if everything wonderful doesn't happen to those people, then the rest of us are doomed. And so we project onto them this wonderful thing, that there are lines of people outside my lab begging me to work in my lab. There are lines of people with big bags of money that are lined up outside, like, please let me fund your research. I don't have to even write up any papers anymore. I just rip out the pages from my lab notebook, send it to Cell, and they write it up and publish it for me. And my grants don't even have to be reviewed. They just give me buckets of money for them. I can tell you that all of that does not happen. I think it's the opposite. I've actually had fewer people apply to work in the lab after the Nobel than I had before.
Because you already got it, so.
Yeah, I'm old hat and over the hill.
There is a question here about how we describe those who succeed. Usually we use words like persistence and resilience, something Lady Gaga talked a lot about at the Oscars. But how do you describe people who have a chance at success, who will deal better with failure? What are some traits you have noticed?
So the answer is I'm not sure, but I want to turn that question around. Okay. I think that the problem is that success is not getting a Nobel Prize. Success is not winning the Olympics. We are successes as how we treat other people, how we're treated by other people, what we do in our lives. Our families are our successes as much as our work is our success. The idea of success being some sort of an award, I'm not really sure. There are people that I have had as friends who I think were astonishing. They were astonishing scientists, astonishing doctors, but they were also astonishing human beings. To give one example, just off the top of my head, Michael Katz, Dr. Michael Katz, was the head of pediatrics. He's emeritus now. He's in his 90s. Emeritus at Columbia Medical School. He was also the head of research for March of Dimes, and he's a consultant still in many aspects. He is a Holocaust survivor. He is an amazing human being, probably one of the most caring people I know. So I think personally, I think he's much more a success as a human being than I would say as a scientist or a physician, even though he was those as well.
Yeah. This is the last question, which came from the audience, and it goes like this. In an age of rapid innovation and subsequently quick obsolescence, in STEM fields, the competition is more intense than ever. What advice do you have for aspiring Nobel laureates? So
2 parts. First, don't try to get a Nobel Prize.
Okay.
And I actually have come to the feeling that the Nobel Prize is a very unusual type of award because it doesn't go to the smartest scientist, it doesn't go to the most productive, it doesn't even go to the best scientist. It goes to scientists who, by their work, change the way science is, some aspect of science is thought of, or how science is done. And those accidental discoveries are exceptionally important for that. Probably the most important discovery in physics in the 20th century was the discovery of the background radiation of the cosmos. It gave substance to the idea of a Big Bang as the origin of the universe. Penzias and Wilson discovered this. How did they discover it? Were they looking for this? No. They were calibrating a machine at Bell Labs. And by calibrating the machine, they pointed at the Milky Way. They got a nice, strong reading. And they pointed it away from the Milky Way, where they expected a reading of 0. They never got the reading of 0. Why? Because there was the background radiation that led to them getting a Nobel Prize. These things that change things, sometimes they're directed. There are some people that say, I want to solve this. And they are able to do it, and I think that's remarkable. But an astonishing number of people write, guy did place cells. Is that Griffiths? Is that his? I've forgotten the man's name. He starts his Nobel speech by saying, one day, instead of putting the microelectrode in the thalamus, I stuck it in the hypothalamus by mistake. That was his Nobel Prize winning work. I told you about Shimamura's prize winning work. These things, you have to be open to these discoveries, but these are things that change the way we think about science or do science. And I don't think you can design things to do that. I'm going to discover something that's going to change the way we do this. The discovery of H. pylori as the cause of ulcers. So I'm not sure to do that. How can one work towards being the best scientist they can, working on that? You have to work. You got to put in the hours. These accidental discoveries only come if you do experiments. If you don't do the experiments, you can't get the results. So it's the old Wayne Gretzky line, you miss 100% of the goals you don't take. So I think that has to be a part of it. Yeah.
With that, we have to close, right? And I want to thank you so much.